Good afternoon. Good afternoon, everyone. It's a pleasure to welcome you to this fall's Distinguished Faculty Lecture, the 59th in this series. So first, a little bit about the series, and then I'll spend time introducing our Distinguished Faculty Lecturer. First, let me start by introducing myself. My name is Charlie Reardon. I have the distinct pleasure of serving as Provost here at Hofstra, a position that I started in July. So like some of you, this is my first semester, and many of you, most of you have been here much longer. But it's a great pleasure in the role of provost to engage with our faculty and students, and so I'm really excited to hear from Javier this afternoon about his work in the Department of Biology. So as I mentioned, this is the 59th Distinguished Lecture. The lecture series started in 1981. And since the year 2000, with the exception of a hiatus during the COVID pandemic, since the year 2000, there have been 2 lectures each academic year, one in the fall and one in the spring. Traditionally, during that time, the fall lecture has focused on scholarship, and the spring lecture is focused on innovations in pedagogy. So today's speaker is Javier Izquierdo, who's an associate professor in biology in our Hofstra College of Liberal Arts and Sciences. Javier holds a Bachelor of Science in Biology from Case Western Reserve University and a PhD in Microbiology from the University of Massachusetts at Amherst. He did his postdoctoral work as a research scientist in a Department of Energy-funded Bioenergy Science Center, both at Dartmouth and North Carolina State University. And then prior to joining Hofstra, which he did 8 years ago, Dr. Izquierdo was active in the industrial sector and had a role as a bioenergy program manager at the Center for Agriculture and Environmental Biotechnology at RTI International. So a very broad background he brought to Hofstra to invigorate our classrooms and our research laboratories. So his research lab explores the metabolic diversity of microbes and their applications, and his group uses classical microbiology techniques, as he and I were just talking about, which are advancing literally month by month in terms of the tools that are at the disposal of microbiologists to solve increasingly complex problems. And his particular interests include the role of microbiomes in promoting plant health and soil environments and the sustainable production of biofuels and bioproducts from agricultural waste. For those of you who may be less knowledgeable or don't spend every day thinking about microbiomes, first of all, you should spend each day thinking a little bit about microbiomes. The microbiome, just to put us all on the same page, if you will, is a community of microorganisms that include things like fungi, bacteria, archaea, and viruses that exist within a particular environment. In the past several decades, we have really, as a scientific community, begun to understand their roles in topics ranging from human health to energy production to agriculture. And to give you one example, but happens to be my own personal favorite example, and I won't go into the details, but I'll encourage you to look up an Australian physician named Barry Marshall, who won the Nobel Prize Physiology and Medicine in 2005. Fascinating story. So Marshall is credited with understanding and developing an understanding of the link between Helicobacter pylori, a bacterium, and peptic ulcer disease. Before his work, people who grew up in my generation thought there were a variety of causes for peptic ulcers, none of which had to do with bacteria. And not to tell you the whole story, he used himself as a subject in his own research, and at one point drank a vessel containing Helicobacter pylori. I'm not recommending anybody take that approach, but read a little bit more about his work in that regard. So a lot to be done in terms of human health, but you're going to hear more, I think, today about the role in renewable energy and in agriculture. So the lecture today is entitled Developing Microbial Solutions to Complex Problems, One Microbe at a Time. And the last thing I'll mention before welcoming Javier to the stage is to let you know that in the classroom setting, you can find this on YouTube, as I did, he does take the opportunity as a talented musician to bring a guitar or harmonica to class. He's told me he brought neither today, so we'll have to encourage him to follow up with that. And I think the other aspect that strikes me in Javier's approach to educating the students here as Hofstra is he likens scientific discovery to either a Harry Potter or a Game of Thrones journey. And so maybe we'll hear more about that as well. So please join me in welcoming Javier.
Thank you, Charlie. Thank you very much for being here, and thank you for that lovely introduction that went into, I thought I had gotten away with it without any stories about the guitar until we got to the very last paragraph of the last page. Yes, sorry, no guitars today, and no Harry Potter, and no Game of Thrones, but hopefully I have a story here that is enthralling enough for all of you. That's the goal, and that's what we'll be talking about today. I don't know, aside from saying thank you, I don't know, how do you thank for being part of this esteemed group of colleagues that have been in this series of distinguished faculty that are called distinguished faculty? So I'll just say thank you. But aside from saying thank you to the community that that put me here, I want to thank. Is this working? Let me see. Yes, it is. I want to thank the people that actually make this possible, which is this is usually the slide that we leave for the very end, and because a number of my students are here, they know that I'm going to be running out of time when I get to the last slide. I'm putting this slide on purpose right here at the very beginning so that we properly thank the people that do the work. These are all the students that have been past and present in the lab. You'll see work that they have done throughout the presentation. They'll be making appearances here and there, so you'll see some of their faces all over again in some of the slides. Everything that I do is, I love what I do, and it's exciting. That brings me to work every single day and sometimes during the weekends. But it would not get done if it wasn't for all these people that have shared this interest with me throughout these past 8 years that I've been here at Hofstra. These are all students that have found what we do in the lab exciting and keep me going. So a big thanks to them, and also big thanks to, we've gotten funding from NSF. This is a grant that I received from the Plant Biotic Interactions program, and we've also received funding from the Department of Energy by way of sequencing through their Joint Genome Institute. We have also received funding from HCLAS Faculty of Research and Development grants. Those are huge, hugely important. So big thank you to all the people in administration that have worked really hard to bring those back, keep them in there. Big thank you to all the colleagues that spend time reviewing those proposals on the regular basis and fund great projects all over Hofstra, including some of the projects that we do in my lab. And as well, I'm going to mention a little bit of our collaborators throughout. So we have collaborators in Fire Island National Park, particularly Jordan Raphael, who is a Hofstra alumnus, and Kelsey Taylor. We have had a lot of contact with local communities in some of the work that I'm going to be presenting. So the towns of Oyster Bay, Babylon, Long Beach, and the village of Saltaire will be featured in different places throughout the talk. And we have collaborators at Dartmouth College, Dr. Ever Holverda, who has helped us with a lot of the metabolite analysis that you'll see, Dr. Larry Smart from Cornell University, and Dr. Bianca Charbonneau from University of Pennsylvania. Good. Now I don't have to run through that at the very end. I've properly thanked everybody, and we can jump into this. So I'll go back to this slide at the very beginning that has, you can see a lot of microbes in there, and some of them are from different species, some of them are the same species. Very complex picture of a very tiny, tiny scale. And this is a little exercise that I like to do. And if you have done this before with me, which some of you might have, you are not allowed to play. That includes students that are in my classes, which I see you, you know who you are, you cannot talk. So here's a sample of sand from the beach that me and my family usually go to, Tobe Beach. It's exactly 1 gram, I weighed it. Quick question for everybody. How many microbes do you think are here? Oh man, this is like being, I just came from class in which every time that not every time, sometimes when I ask a question, I'm met with this strange silence. All right, let's try this again. Also, microbiologists cannot, I see a couple of you in here, you cannot answer that. I'm just asking for an approximation. It doesn't have to be a perfect number. 10, we have 10 million in the back. 5 billion. So let's make it a multiple choice. So we're in the million or billion range. Billion. I like that number. So this is a classic multiple choice question that I would put in an exam. There's 3 possible answers and then one that looks completely ridiculous, and sometimes people pick the ridiculous one, but that's okay. There's 3 good possibilities. We have billion. We have somebody going for billion. We have a lot of people going for billion. You guys have been paying attention, or to whatever micro class you were, or you read ahead of time. I don't know. That's about how many organisms we have in here. So somewhere between 10 and 100 billion. That would be more in a common soil. This is going to be closer to the lower range. So about 10 billion bacteria and archaea in here. We're not counting fungi. I'm not counting other larger organisms. I'm not counting eukaryotes, which some people in here will be insulted with, and they will be right to feel that way. So I'm just talking about bacteria and archaea in here. How many species? I don't know exactly how many because I've counted. I've looked into this. We've processed a lot of these samples of soil. So that's a lot of organisms, but how different are they? How many species? 40,000. I like that number. It's a little bit high. It's in the thousands. So we have thousands of species in here. It's in the lower numbers, so it's not as close to 40,000 as I would like it to be very impressive, but it's somewhere between for this particular sample, 4,000 and 7,000. Richer soil, your garden soil is going to have more of those species, and it's going to have probably twice the number of, or not, 10 times the number of organisms that this would have. So more organisms, twice as much diversity. And in that diversity, this little sample, just this that I have right here, I have organisms that are different. We're talking about 4,000 different species. So we're talking about organisms that can be as different as you and I are from a fungus, as you and I are from a plant. There are organisms in here that carry out photosynthesis, even though it doesn't look much like it, but they are in there. There's organisms in there that can take 80% of the air that is in here, nitrogen, and fix it and turn it into ammonia that goes into amino acids, that go into proteins that can feed other organisms. So there's also organisms in here that are behaving a lot like you and I are. They love sugar and they eat a lot of it, and then they make a lot of CO2 out of it. Very, very, very similar in terms of their metabolism, except that they're microscopic and unicellular and have done a couple of other things in their evolution than you and I have done. So broad range of diversity, and something that we do in my lab is try to figure out what that diversity is. So try to understand that complex system, because we're talking about all these different species. So how do we look at those communities and try to understand them? Luckily, with the tools that we have these days, we can sequence specific genes, and the sequencing technology, DNA sequencing technology, has gotten so good at giving us a picture of what we know there, that exists out there, that we can look at a complex community like the one I'm showing on the far left as a cartoon made of Lego, and we can use molecular tools to analyze that community and get a good census of those 4,000, 7,000 species, and that's how I know how many are in here, because we have looked multiple times. The other part of the work that we do in my lab is trying to understand those microbes a little bit more closely. So breaking those communities apart into pieces and looking at their building blocks, and those are those isolates or microbes that you can isolate from the whole community and grow in the lab. A limitation with that is that you cannot grow everything that is on that community. So some of those organisms are going to remain in the community without us being able to cultivate them. And there's a whole bunch of them that we're going to be able to study very well, and a lot of our understanding of that community and that environment is going to be based on those organisms. But you can still learn a lot from the community by looking at the genes that are present there, looking at the possible transformations that they make, and that's also something that we can do with molecular tools. And the other part that we're trying to do in my lab as we try to address some of these big questions is reconstruct those communities. So bring those pieces back, any organism that we get from those microbiomes and microbial communities, bring them together into what we call reconstructed communities. See what happens when you put them back together and how they interact with each other. Are there negative interactions, positive interactions, null interactions? What do they do that represents the original environment and where they came from? So the talk today is going to have two big parts that essentially describe the two main projects in my lab. Two main projects that try to address these big questions with microbes that we are trying to sample from all over the place, and inquiring a little bit, not so much into the microbiome, the human microbiome, but microbiomes associated with other environments and with other systems that don't have to do necessarily with human health directly, but maybe indirectly. So one of the questions that I'm setting out to, or that we have set out to ask, is how to grow crops in lands that are not suitable for agriculture. That's a sustainability problem that is already, it's not coming, it's already here. And one of the solutions for that a lot of companies are trying to use microbial solutions for that. So the story starts in an unexpected place, because I'm talking about growing crops, and I'm talking about lands that are not suitable for agriculture, and we're going to start in a land that is definitely not suitable for agriculture with a plant that is not a crop. So this is beachgrass, or American beachgrass, which is, the scientific name is Ammophila breviligulata. You'll see that in a couple of places. And after we're done with the talk, I'll quiz you and try to get you to say Ammophila breviligulata 3 times in a row and see how that goes for everybody. I cannot do it, so it'll be very impressive if you can. This is a plant that we don't eat. Humans don't eat. Some herbivores do eat, but it's a very sturdy plant. It's not a good plant for any herbivore's regular diet. It also grows in a land that I wouldn't use to grow anything. You don't want to grow any of your vegetables in there. So it's a weird place to start asking this question. But it is a plant that has an important ecological role. It has these adaptations on its root system and these adaptations on its leaves that allow it to trap sand on the beach. And by doing that, it's known as an ecosystem engineer. It forms those dunes. It traps that sand and increases the height of the dunes. And as it's doing this, it's creating new environments. So now you have places where there's carbon fixation, nutrients being more abundant than they were before when there was just sand, and new plants can colonize these areas, birds can nest, and you see how this sort of balloons from there. And also in terms of the stability of barrier islands all over Long Island, but all over the Atlantic coast, and also you can see it in the Great Lakes area and in other places in the world. It is a plant that protects those communities as well, right? So it has also an important societal role, the role of building those dunes. And it has its enemies as well, right? So there are events like this is a picture from Tropical Storm Sandy. There's other storm bends that are becoming more and more common. And this is it has a big effect on the stability of these dunes and the stability of these barrier islands and these ecosystems, but also on the role that these barrier islands play on protecting our communities. So you'll see a lot of places, a lot of towns like my town, like Massapequa Park and the town of Oyster Bay, how we will go through these replanting events. I usually take students to those events, and we haven't done them in a couple of years because of COVID, but it's about time we got back. But these are replanting events that happen every year, particularly early in the spring, and trying to bring beachgrass back into these dunes and get it growing again. And for us, the microbiologist that I am, one of the interesting things that I find about the beachgrass is that a lot of what it needs for that stability is based on this complex root system, right? And all the microbial interactions that it might have with organisms that we don't know much about. So the publication record on beachgrass is very, very limited. There's a couple of species that have been studied, and not much that is known about the microbiome that is associated with the plant, the role of that microbiome in the health of the plant. So we've started looking. And this is a picture also, kind of like the opening picture is from scanning electron microscopy facility in our department that allows us to take these more detailed pictures of microbes that you can also spend time coloring like I have and see here in all their glory. But you can start seeing some of the morphology. This is a root from beach grass, one of those thin hair roots that you're seeing here. A little bit of a close-up that allows you to see some of the different morphologies and some of the different organisms that are there. And from knowing what happens with other plants, we know that microbes associated with the root of the plant play a huge role in bringing in nutrients to the plant and also in some cases producing plant hormones that help that plant grow. And we set out to use this approach that I introduced earlier on to understand that ecosystem, right? That this underexplored plant that has some important ecosystem roles and societal roles that we know very little about what the microbiome does for it. And so we set out to answer some very basic questions. Who is there? What organisms are there? What makes that community, both in terms of the microbiome, using molecular tools, using DNA to see everybody that is in there, a whole inventory of who's in there, and then seeing what we can grow in the lab, looking at those isolates. And learning more about what they're doing. What is it that they can do for the plant, if they're doing anything, and see if we can relate that back to the ecosystem. And NSF thought that this was a good idea, of something that was worth pursuing, so we've been doing a lot of work in that area. We've been sampling all over the southern shore of Long Island, a lot of sites that have wild beach grass, also replanted beach grass in different states of health. And this has allowed us to see, get a better picture of what's happening here. And something that we've been doing in every sample is looking at different environments in the sample. So we are not only looking at the soil that surrounds the plant, but the soil that is directly adjacent to the plant, this area called the rhizosphere that you see depicted up there, as well as the inside of the root, right? What makes the community of the microbiome that is inside of the plant compared to immediately outside, and then a little bit further away, which is what we call the bulk soil. And the first time that we looked at this, or the first couple times that we've been looking at this, we did this big survey looking at a whole bunch of places. So this is 3 different samples, and I'm going to move a little bit. I think I can project enough to hear. Sina, you can hear me? Perfect. So here we have 3 different samples, or 3 different locations. These are all on Fire Island. And I don't have to convince you too hard to look at, so this is bulk soil, rhizosphere is the soil adjacent to the root. This is the inside of the root. That the soil that is adjacent to the root looks a lot like the root, the inside of the root, right? So in terms of the community composition, that's what we have in there. The community composition here, so just to give you an idea of what that community composition is like. So for each one of those samples, we're talking about tens of thousands of DNA sequences that we have matched with something on the databases, right? That gives us an idea of the taxonomic place that they have in the bacterial tree of life. So these are only bacteria. Sorry, no archaea. That will be a whole other figure of these bars that everybody will be looking at. He has another one of those figures that we're going to look at. So we're not going to do that. But in terms of the bacteria that are present, this gives you an idea of taxonomically where they belong. And we can see that taxonomically there's you can pick your adventure, right? So if I ask any of my students or microbiologists in the audience, they will pick their favorite organism and oh, what's happening there with the alpha proteobacteria? I love those guys. But we're not going to explore that just yet. We're going to go into more detail looking at these guys in a more finer or much finer taxonomic level. But you can tell already that the microbiome is structured, right? The soil directly adjacent to the root is very different than the soil that surrounds it, and much more closely related to the organisms that are inside of the root. Makes sense as an environment. But it also, every single time that you go to the beach and say, do not step on the dunes, now you might have a little bit of a microbial appreciation for what stepping on the dunes does to that environment, right? Disturbing that environment actually does something to the soil. You end up mixing that bulk soil with that rhizosphere soil, and that affects, just in terms of the microbial community, it affects what we're seeing in there for the plant. We went a little bit deeper and looked at the genus level, which is what these molecular tools are really good for. They're not fantastic to letting us know a specific species. This is a perennial problem for microbiologists, defining what a species is, but a genus with molecular tools we're pretty decent at. And this is organized from organisms that in Tobay Beach and Cedar Beach, where we sampled sites that had plants that were in different states of health. So what we're calling healthy versus unhealthy plants that were successful growing there versus plants that were not. And when we looked at the composition of those organisms, we had hundreds, close to 600 different genera. These are the ones that changed most significantly. So we're simplifying the picture here by looking at the ones that changed, that had the most significant change. And we're seeing in blue the ones that were more abundant under healthy conditions. So those are the ones that are at the very top of that picture, and some of them were found on both sides to be across the board in soil, rhizosphere, and endosphere, every single microenvironment across the board, more abundant. And then at the other opposite end of the spectrum, you can see down here at the bottom, the ones that were more abundant in the unhealthy plants. And not to make you squint too hard, I have a Cliff Notes version of this slide that addresses also some of those top organisms. So those are the ones I'm highlighting here: Mucilaginibacter, Verrucomicrobium, and Phytoplasma. Those are the genera that we found that were present in healthy plants most prevalently in all soil microenvironments. But there were some that were also in environments that we should care about the most, which are the rhizosphere and the endosphere. And that included a lot of organisms that were also involved in nitrogen cycling. Makes sense? This environment is very deprived in nitrogen, so organisms that fix nitrogen are going to play a role in that. Organisms that cycle some forms of nitrogen that might be present in the forms of salts are going to be useful in that environment as well. So that includes organisms like Nitrobacter and Rhizobium. Makes perfect sense so far in terms of the organisms that will be associated with health. The ones that are a little bit puzzling are the ones that are present across the board, because two of them we really cannot grow in the lab. Verrucomicrobium we kind of can grow, and by we I mean the whole microbial community or the whole microbiology community. Not me personally. I've tried and failed miserably trying to grow this organism in the lab. It's very fastidious. It has very specific growth conditions, very specific media conditions. We haven't been able to grow it. Phytoplasma, nobody knows how to grow in the lab. It's an organism that is an internal organism of the plant, and you cannot separate it from the plant. So you would have to have a medium in the environment that is exactly like the plant environment, which we cannot do yet. So everything that we know about Phytoplasma as an organism is based on molecular tools. And that leaves Mucilaginibacter, that is an organism that we can grow in the lab. And we know a couple of tools of how to grow it. So we're going to put a pin on that because you can tell that we're definitely going to come back to that one because it might be worth exploring. So of the three top organisms, it's the one that might be worth looking more into. And within the organisms that were at the bottom of that graph, the organisms that were more associated with these plants that were not doing that well, we found a whole bunch of root actinomycetes that are found in desert soils. So organisms that are not really, and by desert soils I mean in the middle of the desert, not associated with a plant. There's plants that are successful in the desert, and if we had found organisms that were associated with that desert plant, that would have been cool. But we're finding organisms that are associated with essentially no life. They are out there to live with themselves. So we have continue to look into, so we didn't stop at this survey all over these sites. We have looked at this not only in terms of the whole of Long Island, we have actually gone into New Jersey as well and looked at beach grass over there, and we'll tell you a little bit about it later. We have also looked at this over time. So we had a long-term study in which we looked at the progression of dunes as they become bigger and bigger from the moment the plant starts colonization as it starts making dunes that are taller and taller. So that's what we call primary, secondary, and tertiary dunes that are being shown here. So primary is a dune that is in the early stages of formation. This is something that just started getting formed that particular year, and it's at the level of sand where it's meeting the ocean. But that primary dune, by 2-3 years on the road, it will start looking more like a secondary dune that you see in the back. So we've done this over time, and we have looked at organisms that are prevalent in the early stages of the colonization of that dune, and we found again organisms that fit the same descriptions that we saw before, some of the same genera. So we found Mucilaginibacter again, we found the nitrogen-fixing Rhizobium as part of that. This was the first time that we had done 2 big studies that match each other, so that's kind of exciting. This is one of the students responsible for that work, celebrating. He was not celebrating that we had found that out. He's in the middle of sampling here, but it's the same idea. This is Josh Pimentel, who did a lot of work in that area. And he also, for that work, he received this prestigious fellowship from the American Society for Microbiology, the Undergraduate Research Fellowship, this national fellowship that is awarded every year from the society. And it paid for his research for summer session, but also travel and lodging for the general meeting for the American Society for Microbiology. And they had like a whole week-long workshop ahead of the meeting for professional preparations. He had a fantastic experience. I wish I would have had it as an undergrad. That never happened for me. I'm happy that it happened for him, and that this picture that you see here is essentially his whole experience through that grant and the trip to the American Society for Microbiology meeting. In a recent study that we published last year, we went a little bit further. This is a study in New Jersey, so we left Long Island for a little bit to study with a colleague in the University of Pennsylvania, who has a bunch of sites over there, the comparison between an invasive plant, Carex kobomugi, and beachgrass in places that have been blown by storms. And in both plants, we found some commonalities in terms of the kinds of organisms that they recruit. So they're plants recruiting organisms in a barren area. But we found that for Ammophila, some of the same organisms that we were talking about before, again, are popping up. So comparatively, when you compare it to other plants and plants that Ammophila is competing with, we are finding some of those same organisms. So that's the type of microbiome level studies that we've been doing in the lab. And I promised you that we were going to talk about building blocks, so let's talk about some of those as well. We've been assembling a massive collection of building blocks or individual isolates in the lab since this work started. This is Sherry Saslow, who was one of the students that started that work, the work that I showed you earlier on in terms of the microbe community. She also participated a lot in the work of initiating some of the isolations, but this has been work that has been done by a variety of students that you're going to see listed in the next few slides. And some of the things that we can do to look at those microbes, those possible microbial solutions, those building blocks, is see, for example, what kinds of plant hormones they make. And we have a test for looking at auxin, indole acetic acid, that allows us to see some organisms that make a lot. And you can see these organized in here from some that make ridiculous amount, to be very scientific about it, of indole acetic acid. Anything above 100 is considered to be an organism that makes indole acetic acid that will have an effect on growth of roots with the plants that they're associated with. So these are the ones that we listed that were 27 isolates that had more than 100 micrograms per mil of this particular plant hormone. There were, of course, a whole bunch of them that are the other sad, that little bar at the very bottom that looks very sad. Those are the others that made just okay amounts. And I'm seeing some faces of disappointment from some of my students who are measuring those activities in one of my classes right now. And they're thinking, that's where our microbes are. There are other things that microbes can do, and you know this, right? So let's see what happens. The experiments are not done. Don't get too disappointed. We can do all kinds of other cool tests in the lab to see what microbes are doing. This is a test that allows us to see if they're sequestering any iron from the surrounding environment. That's what you see in those halos that are on this plate. We can also see if they're making interesting products that kill microbes around them. So this is a fun animation that I like to show. And if you're paying close attention to what's happening on that plate, that's a whole bunch of different organisms growing together. Some of them get along, some of them don't get along. Some of them, if you look closely, are dropping the equivalent of what is an atomic bomb in different places. So these guys right here that are right in the center of those areas, they're producing some sort of antibiotic that is clearing the area around them. And maybe you can see it now that I've dropped atomic bombs and drawn little circles around it. That might be a little bit clearer. So you see these organisms that can produce antibiotics and might be doing that to be in warfare with the organisms that are around them, but also some of them might be doing that in terms of protecting organisms, the organisms that are associated with, right? That could be the plant. So with this collection that we've been assembling, some of it has been, let's go out there and collect whatever we can collect and test it, right? Test it for hormone production, antibiotic production, so therefore production, other metabolites that might be out there. We have done a little bit more of targeted isolation as well. So this is a student in my lab, Arjun Kumar, who, with our knowledge of the microbiome, that mucilaginous bacteria is important. We have multiple studies in which we know that mucilaginous bacteria is an important component of healthy microbiomes, early microbiomes, and microbiomes of the plant compared to other plants that might dictate something about the success of this plant. So we've gone out there and just tried to hunt for mucilaginous bacteria. There are specific types of media and specific tests that allow us to look for it. And this is work that he did also with the help of a high school student, Dennis Sinar, from the Hofstra program during summer high school experience for Dennis. And we have had a couple of those students in the lab. And she did fantastic work. She complemented the collection that we had with that Arjun was assembling and helped us isolate some of these organisms and test them and get a good sense of what kinds of compounds they were producing. So every single thing that you see listed here as an organism with several dots right next to them on this phylogenetic tree shows a mucilaginous bacter organism or mucilaginous bacter-like organism that is producing something interesting. So organisms that we're testing a little bit further, and I'll show you a little bit about them. So in all of these microbe hunting and isolations, we've been able to create this nice collection and start picking some organisms that are very interesting. Some of them I'm going to tell you a little bit more about before the end of today. But we have mucilaginous bacter that you see there in pink. That's also on purpose because they make these beautiful pink colonies on agar. Sphingobia AW4, I'm going to tell you a little bit more about in a second. But we have also organisms that fix nitrogen, like that Herbospirillum and that Penebacillus that's taking nitrogen gas and making it into ammonia. We are in the middle of hunting for more of those organisms in the lab, and we have a student that is in charge of that. We have Pseudomonas that also makes a lot of hormones, and we're seeing what else does it do for the plant. So we have all these organisms in different categories, a little bit trying to get as much diversity in there to see what we can assemble together of this microbiome. And part of this has been also enhanced by help from the Department of Energy and their Joint Genome Institute that has sequenced for us at no cost to us other than providing the sequences, providing the organisms, 48 genomes of these isolates. So, and we have all those genomes sequenced, and we're starting to do a little bit of work with all of them. There's a lot of data in there to get through. I'm going to tell you a little bit about one of those organisms right now. So this is a paper that we just published this year about AW4, a strain that we love, and I'm going to tell you more about why we love it, not just because we have published the genome and we found cool things in the genome, but also it does a lot of cool things for the plant. In the genome, we have found out that the organism has all these specific operons that no other Sphingobium or anything that we call Sphingobium has. So, for example, these are a set of genes that allow this organism to use fructose as it's being secreted by the plant as a signal molecule or as a molecule to recruit those organisms. There is nothing in there. The reason why there's two other Novo Sphingobium or Novo Sphingobium is a different genus. There's nothing within the Sphingobium genus that has this operon. And this is just one example of the different types of molecular capabilities that this Sphingobium has to use sugar specifically released by the plant. When we look further into what Sphingobium has, so along with those operons for sugar utilization, there's other sugars or monosaccharides that Sphingobium has. So compared to other Sphingobium species that are shown right next to it, so those are starts with the closest relatives at the very top, and then it goes on to less and less closely related Sphingobium species. You can see that in certain categories, Sphingobium is the champ, right? So it has more genes involved in C1 metabolism than all of its closest relatives. It has more genes involved in production of plant hormones than all of its closest relatives. It has more than most in terms of osmotic stress, which also makes sense in terms of the environment that we got it from. So that makes it an interesting organism for looking at what it's doing with signals from the plant in terms of carbon metabolism, signals to the plant in terms of those plant hormones, and surviving in rough, difficult, salty, and dried environments where they would be going through these levels of osmotic stress. So we have tested these organisms and others with plants to see, I've been showing you a lot of, okay, this is the organism, this is what they do, but what happens when we actually test it with a plant? And this is an example of that. So this is Sphingobium growing with Arabidopsis plants. These are plants that you can grow in the lab, on the bench, on plates. And of course, you can take them to, it's a plant, so it it's meant to go into a pot. But these are experiments that you can do relatively quickly with the plant and measuring how the microbe is affecting the growth of roots. And we are seeing, you can see that all the organisms that are at the very top inoculated with the organism. You can see residual coloration in there for sphingomyia attached to those roots. How those roots are much longer than what happens when you have a control without any sphingomyia in it, or sometimes even with just sterile media. So those are all the other controls that I'm showing at the bottom. So that's a good indication that it's helping this plant model grow. So we have taken it further than that. Aside from we're growing in a lab, we're growing it also in this greenhouse facility that we have on top of our building in Gittleson. And we're testing these organisms with other plant models. So one example of that is testing it on wheat. So we have tested AW4, we have tested other organisms as well, other isolates. This is SJZ 102, that not only a lot of the organisms that we have either promote root growth or promote upper leaf growth, and this is an organism that promotes both. And we're still trying to figure out all the details why we have the genome. We're working on what is it that we know that some of the compounds that it makes, but we're still dissecting the genome to figure out what else is it doing to produce these levels of differences that you're seeing here, specifically on wheat. And I did promise you to sort of bring this back together, right? To get all those isolates, and as we're still trying to understand them, to put them all back together. And that's a little bit of where the future of our work is going, and the future is kind of now, right, for us. We're doing this work that Amber Jordan is doing for her master's thesis in my lab, putting these microbes together, bringing them as reconstructed communities, and seeing what happens. And she's done the same kind of analysis that I showed you before of looking at the community composition, which is what you see on the panel on the right. How does the addition of these microbes as a community affect the whole community, right, and affect the whole plant? And something that she's been finding is that under certain conditions, under sand, not vermiculite, which is a different substrate to grow the plants in, but growing them on sand, that those organisms, if you look at the y-axis, there's the percent change of how much those organisms grow in the presence of the plant is massive. Like after we add them to the plant and in these pots, the plant is promoting the growth of these organisms. In particular, the one that is the most affected is the one that we see on the far right, OTU14, which is AK15. That's shorthand for a mucilaginous bacteria organism. So the one that gets the most benefit of this is a mucilaginous bacteria being promoted in terms of growth by the plant. We're also trying to figure out, we're doing a lot of molecular work to understand what interactions are happening between these microbes as we put them together. But that's ongoing work, and that's what we're living it in as the future. But the future is still looking bright. We have, you might have seen Amber in the news last year. This is when her work and her poster won the first place award at the Regional Tribeta Convention. That's the Biology Honor Society. And another member of the lab, Juliana Dolinsky, got the second place that year. So Hofstra made like thieves, and we left that place with first and second place in terms of the posters. And Amber and Luciana were featured in the news, as you can see in here. The future looks also very futuristic for us in this project. This is, I hope I'm not jinxing anything, but it's not that many times that I'm going to give a distinguished lecture, a distinguished faculty lecture. This is probably my only time, so I'm just going to jinx myself, and that's okay. So we have a proposal with Bayer, and Bayer is supporting work at University of Arizona in Biosphere 2. Has anybody heard of Biosphere 2? Yeah. So Biosphere 2 is this fantastic research facility that has all these different biomes in there. They have their own ocean with their own coral reef, which sounds incredible. It has its own rainforest. It has all kinds of biomes from the Earth. But they also have this very cool thing. They have this thing called SAM, which stands for Space Analog for the Moon and Mars. Bayer is funding projects that allow you to test in an analog environment to the moon and Mars to test microbes that can help plants grow. So our proposal has made it through 2 rounds. We are on the finalists. Wish us luck. I might have just jinxed us, but I think it's very cool, and I'm geeking out because I might get to be Matt Damon in The Martian, and I don't know where this is going, but it might be very cool. I don't know what's going to happen. So, but that's something that we have coming up and that we have in the works. And something else that it's more close to Earth and more realistic is this possibility that we have with Joint Bio, which is pun intended, a joint venture between Bayer and Ginkgo Bioworks in Boston, who are looking for microbes to help serve as biofertilizers for specific crops. So we also have gone through 2 rounds of discussions with them. I had an interview with them over Zoom 2 months ago, and we're waiting to hear what's going to happen with funding for these projects. We are feeling pretty optimistic that they might choose some of our microbes that we have on the pipelines to test on some, to make it a real testable biofertilizer that they want to also develop as they call it chases for fixing as a car that you're going to bring in new tools and genetic tools to modify and add other features to something that is already really good. So we'll see where that goes, and we'll see where that takes us. All right, we've gotten through part 1. Part 2 is shorter, don't worry. We have this other part of the lab that works on biofuels. And the question that we're trying to address there is how can microbes help us make renewable biofuels for the future? And this goes to, I have used this picture in every presentation that I've had for close to 12, 13 years at this point. These are my kids when they were much younger. They're teenagers now. I'm not looking at them because they're rolling their eyes at this picture in this moment. I'm not making eye contact. But this is a moment that I loved because it was done naturally by them, and I had to go back inside the house and get the camera and say, stop. Continue. Let's capture this moment, because they were making this or staging this metaphor for what my work with biofuels is. There's many ways of making biofuels, and here's one of the ways of doing that. And they were shoving grass into the tank, or cellulosic material into the gas tank. Fantastic game and a great photo op for me that I continue to use in pretty much every talk that I give about biofuels. And they make it look simple, right? As a game, it's a simple thing to do, shove it into the car and the car is going. But it's not that simple. This is a very complex problem for the biofuels industry. We've been trying to do this for a while. We have come up with ways of doing this that make it sort of efficient, but the problem is it's not just efficiency, it's how does it compete as a process with real oil, right? While oil and oil-generated gas is cheaper, it's still not. This is not going to be competitive. So we have to come up with ways of doing that. And there's parts of this that we understand very well, right? And some parts are obvious. So there's a very straight, clean, direct arrow between sugars and a fuel. In this case, bioethanol or ethanol. We've been doing that for 5,000, 6,000 years, right? We've been brewing for a long time. We know we have a decent idea of how to do that. And we have a better idea now with, particularly over the past couple of centuries, of the science behind it. So we know how to get that part. The tricky part is go from the plant to the sugars, the degradation of those, right? Because that usually takes a long time in the real world. Although there are organisms that are able to do that. And there's organisms that are able to do that in one single step, to break plant material, as complex as it is, with all these polymers that are all embedded onto each other, all interwoven, and then break that into individual sugars that they can ferment and turn into something like bioethanol. The other part where we are very uncompetitive in is the fact that every single process is one straight line. So it would be just the line that you're seeing at the very top, not the diagonal. The reality of a refinery is that the refinery is going to make multiple products, not just one, right? It's not just gas, but it's making gas, plastics, and all kinds of other products that come from oil. So we have to figure out ways of turning that plant biomass in one step from plant biomass to the sugars, to the fuels, but also something else, some other product. Ideally, something like a bioplastic, right, that can take over that part of the market. So this is a very complicated problem shown in a very, very simplistic slide, but I think I gave you the overview of it. So for us, the goal is, can we get there, right, to an organism that can do all those things, those ideal things, or some sort of culture environment, some microbial solution that does that. So this story has three protagonists, three organisms that we've been playing with, three pieces of the puzzle: a cellulose degrader, an organism that can break plant material, a bioplastic producer, and a specialist. And we'll start with the cellulose degrader. So this is an organism that I've been working with for a while, since my postdoc, since I took that picture of my kids taking the car for a ride. And a lot of organisms that are able to do this very well are thermophilic organisms. That means that they love high temperatures. That has to do with how quickly those reactions happen at high temperatures and how quickly they can grow. That makes the process much more effective. The rates of growth are higher under those conditions. This is a compost pile, a thermometer shoved into a compost pile where we're collecting a sample. Notice the temperature. That's not very cool. These are organisms that's where they're going to be at. That's going to be their hotspot, and that's going to be, hotspot is pun not intended, that's going to be where they love to grow. So we've isolated organisms from there that are able to do that. There's organisms that we've studied very well. So C. Thermos Elements is an organism that has been studied for the past 120 years or so. So we know it very well, and it's actually considered to be the model organism for this type of conversion, taking plant material all the way to fermentation products in one step at high temperatures under anaerobic conditions. My boss at the time is a big C. thermosellum scientist, asked me, can we go out there and look for other things that are there? This is your thing. You are the microbial ecologist. You go out there and find things. Are there other organisms that can do it? I was like, yeah, challenge accepted. I'll go. We'll go and find out what we can find. And we found these organisms. So C. clariflavum had just been isolated, and we found a strain that we call strain 42A, but it's a strain of C. clariflavum. It's a variant of C. clariflavum, and that's why we developed an interest in Clostridium clariflavum. They are a little bit different, and you can see here in these bottles. These bottles are loaded with nothing. There's no carbon sourcing here other than switchgrass, which is a biofuel crop. And there's an increase, you can see there in terms of cloudiness, how many cells are growing in there, but also the plant is disappearing, not just because of the cloudiness. This is not some sort of cheap magic trick. Not just because of the cloudiness, but the plant is also disappearing. It's getting degraded better as we move from left to right, from the model organism that people have been studying for a while to the one that we were able to isolate from compost piles. We actually measured it, so it's not a qualitative measurement of degradation. We measured this, and you can see that here, in terms of what is left behind in terms of degradation, there's less left behind by strain 42A than there is by any of the other organisms. And part of that is, and I've published a couple of papers on this, there's plenty of characterization of the organisms. One of the reasons for that is that all of them are able to degrade cellulose very well. But there's other polymers in plant material that are useful, that are made of 5-carbon sugars. So Thermosellum is able to break those polymers, we call that hemicellulose, into these little chains that you see at the very bottom. That's on the far left. Those chains. Clariflavum, the one that people have known so far as clariflavum, it's breaking down to individual sugars, but is also able to do nothing with them. Whereas our organism, 42A, is able to do all the breakdown and then use those sugars for fermentation. So it's using something else from the plant, aside from using cellulose, to make fermentation products. And part of the reason why we found that they could do that, this is the first paper that came out of the lab when I arrived here. This is my first 2 undergrads in the lab, Elise and Ken, was because we were able to find this particular section of the genome that has genes that are involved in using that cellulose, using those 5-carbon sugars. So it had this is not unique to this organism, but it's unique to that clade, within this clade of that particular organism. So Clostridium thermosellum doesn't have it. People having spent years trying to engineer that capability into Clostridium thermosellum, and here we are finding that in clariflavum 42A as an organism, something that the organism can do naturally. We have also been doing other kinds of cool things. So this is John Guevara, who is here receiving also the first poster award, a different year than Amber and Juliana. First poster award for the Triveda Regional Conference for his work evolving Thermosellum to withstand higher concentrations of ethanol. He didn't do that by himself. He's getting all the credit. So I'm putting here a couple of people that also did part of the work. Subia followed up the work after John, and Srilu initiated that work as well. John just got to wear the suit and just get the plaque, but we shared, and they know that we shared with them. So that's our organism, the cellulose degrader, that is doing all these cool things, and we're starting to adapt it to do better things in the lab. We have also this bioplastic producer that also came as an accident. So we started looking in other places. I got tired of looking at compost piles, and we started looking in other places. We started a collaboration with the Bronx Zoo, looking at organisms that we could get from the guts of herbivores over there. This is Justin Andasi looking at organisms that we're isolating in an anaerobic glove bag that is a completely anaerobic environment for these organisms, so we can get the right type of organisms. We got samples, scat samples from okapis, giraffes, ibex, all kinds of other herbivores as well. I'm mentioning these three because they gave us some of the coolest results, and the okapi is one of my favorite animals. If you ever get to go to the Bronx Zoo and go through that exhibit, that's a treat. And what they started with looks a lot like what you see on the far left of that bottle on the left. And in the end, after feeding them only cellulose, they transitioned to look a little bit more like a culture that looks on the far right. And even when we look at that culture, it's still a mess. It's still a community. It's still multiple organisms that are sort of inhabiting together. We started with something that is really the ibex or the okapi gut microbiome, and we have sort of evolved this to be a cellulose-specific version of that microbiome, right? And when we looked at this using the same tools that we saw before, looking at every possible organism that is in there that we can sequence, we have found that some of these enrichments, some of these samples were producing a lot of succinate. And a lot of that succinate was also associated with, in the okapi samples, with high concentrations of, or high proportions of this organism, Clostridium thermosuccinogenes, that, as the name indicates, maybe it doesn't, but it will in a second, is an organism that grows at thermophilic temperatures and makes a lot of succinate. It generates a lot of succinate. So we got interested in that organism. We're feeding them only cellulose. The organism is not able to degrade cellulose, but it's doing that in collaboration with cellulose degraders that are some of the other blocks that you see on that graph, right? So it's a co-culture that is using cellulose that gets degraded, and we're producing this bioplastic precursor succinate as part of what these organisms are doing. We were very excited about it. We said, this has to be a brand new organism. This is going to be something revolutionary. And then we got one of our students, Sam Lau, to isolate the organism from the complex culture. And it looks, as his phylogenetic tell you, it's pretty boring in terms of similarity to the closest organisms that there are, right? So that tree just shows you that our organism, SWL2, which is the strain that we isolated, is closely related to two other Clostridium thermosuccinogenes that look exactly the same. We sequenced the genome and we found some differences in terms of certain capabilities. So our organism is able to use 5-carbon sugars also very differently, which makes sense for being an organism that is a little bit in the wild. But one thing that we found out that is kind of cool is this ability of our strain, SWL2, to make, in addition to succinate, it makes a tiny little bit of ethanol. So it makes comparable amounts of succinate in succinic acid that you see listed up there. It makes other fermentation products, things that we're not really interested in. But it also makes ethanol, a tiny little bit, but that's a tiny little bit that is promising. So we have another project of evolving this organism that is going on in the lab right now that is being carried out by Sachi up there to evolve this organism to make more ethanol, kind of like we did with Clostridium with Clostridium clariflavum before, the cellulose degrader. So we have two cool organisms, a cellulose degrader, bioplastic producer that could be improved. Let's see where this takes us. And then there's this specialist that also came a little bit as an accident. This came from an interest in shrub willow. So if you don't know anything about shrub willow, you will know a little bit about this. And if you want to know about shrub willow, you need to talk to this gentleman right here. So this is Dr. Larry Smart from Cornell University, giving a talk to local farmers about shrub willow and the benefits of this plant. So it's a plant that grows in very poor environments. So it's not competing with agricultural crops. It grows in soils that are considered to be substandard for agriculture. So great area to grow a biofuel crop, and also grows very quickly. So we are looking at that plant as a possible biofuel crop that could be degraded by organisms and make biofuels. The problem with it is that it's a woody material, woody plant that is very resistant to degradation. And that's what the challenge is. So Larry said to us, you guys need to figure out a way of making this degradable. Everybody tries to put Clostridium thermosellum in it or something other, and the degradation is very low. And we said, challenge accepted, we're gonna take it. And I should have not taken the challenge, because the reality of it is that we found exactly the same results that people were getting. Thermosellum does very poorly with it. Our cellulose degrader, Clariflavum 42A, does a little bit better, and that's what you see up there on the green. And that probably has to do with the same phenomenon that we've been seeing before of the ability to use those 5 carbon sugars. Mixed cultures that came from the Okapi as well did even better. That's kind of promising. So that means that there might be some synergy in there on top of some of the specific abilities of these microbes to do that, that tell us something about that do something with the plant material. So even though it's 20% degradation, not that impressive, it is better than what exists already in terms of pure cultures, or what we consider model cultures. So we've gone in there deeply and tried to isolate the organisms that are there and identify how the community changes when you put them on this shrub willow as the only carbon source. And we're looking at some of the same organisms that we've been seeing before. So you see those blocks up there of the sequences that we're finding is same organisms that we've been talking so far. Thermosuccinogenies is there, Clariflavum is there, Thermosellum is there, so cellulose degraders, the succinate producer, and that little block at the very bottom that says Clostridium canicola. That's a new organism that we're interested in, because it seems to be the only organism that exists that has been isolated. There's very little work on it, and it can grow on certain compounds, like salicin, that are part of the biomass of salix. That's where actually shrub willow, the scientific name of shrub willow, comes from that some of the biomass, some of the sugars that it has as part of its woody material include salicin. So we've been using salicin as a carbon source. We've been using all kinds of other carbon sources. We've gotten very close. So the bars that you're seeing here, that's what happens to the community or to the proportion of that organism once you add salicin. So we have a way of increasing the proportions of that organism. The problem is trying to separate it from everything else. So that's a task that has fallen right now on one of my students, Mike McKenna. He's done a lot of progress finding other carbon sources that might enrich for that organism, and we think we're getting very close to getting that organism out. So that at the end we can get to this punchline, right? Get these organisms back together, the specialist, the bioplastic producer, and the cellulose degrader, and have something that resembles these microbial biorefineries, something that can take cellulose, or even better, willow, right? A plant that is growing on all these substandard environments, and be able to make a biofuel and maybe a bioplastic out of that. We're getting to that point where I can be Captain America and say, Avengers Assemble. We're not there yet, but I can almost taste it. We're almost there. And we've started trying to do that. I cannot wait for many, many years of watching these movies to get to that final movie in which that actually gets said. That's the same situation here. We're trying to skip the line and then start putting some of these cultures together. So there's this project that has been going for a little bit, trying to see what happens with Clariflavum and Thermosuccinogenies as co-cultures. And we've been finding that they actually can produce succinate and ethanol together. So the biofuel and the bioplastic precursor from cellulose under specific conditions. So under some conditions they do better, under some conditions it makes more ethanol, under some conditions it makes less succinate. But overall, they do better than they would as individual organisms. So that's very promising, and we're continuing to do that work. So that brings me to the end of this. I'm back to this figure that I opened with. So hopefully now that when you start thinking about microbes, and I had a conversation early on with Fred, I think, about every time that people mention microbes, people immediately tend to think about disease and the medical implications of microbes. But there's so many other things that microbes do, and that's one of the things that we're doing. And maybe next time that you're at the beach and looking at the sand, you're thinking a little bit about these bugs. Maybe the next time that you're at the zoo and looking at what the okapi is eating and defecating, you're thinking about some of the potential cool things that could come out of that, right? As opposed to just walking the other way. Anyhow, I'm going to thank my lab group one more time, and I'll be happy to take questions from all of you. Thank you so much. So I think there's microscopes around, so
Hey, great presentation, Javier.
Thank you.
I was just curious. We all live in the suburbs. We are surrounded by front lawn or back lawns, and everybody is preoccupied by using these nasty chemicals to kill these weed invaders which come into these ecological systems. Is there any way that such DNA-based tool can actually be used to fight weeds with microbes? So rather than we put carcinogens in our backyards, we'll be able to find out which microbes are going to be to the detriment of certain weeds that are invading, and we'll be able to eliminate them with.
There are so many companies looking at that right now. So every company that is looking at a microbial solution is looking, or a microbe as a biofertilizer is looking at also microbes that can do that target specific weeds, target specific, and the idea would be similar to what we're doing here, right? That you're looking for microbes that can be reintroduced into that environment that are part of that environment. So that's kind of the idea of going to beach grass and getting isolates from beach grass that can be returned to help beach grass grow better. Right? A lot of people are looking at native microbes of these environments to modify them and bring them back and do it instead of making a modification on the plant or adding toxic chemicals that you are doing this, right? Bringing one of those organisms back. Yeah, absolutely something that multiple companies are pursuing.
Okay,
yeah.
Thank you.
Dr. Riggle.
So the beginning of this at Beachgrass, again, healthy dunes, unhealthy dunes. You showed pretty clearly different bugs populate those dunes.
Yeah.
I'm interested in the transition states between them, like the decline to an unhealthy state. In particular, what role might bacteriophage play in that process, right? Because you can imagine an outbreak of the lytic phage could decimate the population, give time for others to come up and occupy that space. What do you know about that? And if NSF would throw a little money your way, could you help unpack that?
Are we thinking about a co-proposal?
Well, it's an idea.
So Dr. Riggle is talking about viruses that bacteria deal with, just like you and I have to worry about certain viruses, about some more than others. Bacteria have their own viruses as well that also play huge roles in the control of those populations. So, and to tell you the truth, I don't know what exists out there in terms of phages that are associated with these shifts in population. That's a great question and something that we should talk about more. Yeah. Could you simulate it? We could try that, right? But that depends on how easy it is to get some of these phages from this environment. It have to be phages from the same environment, and you and I know how tricky that can be from environments that are rich in phages, like a pond or soils that are rich in them. That's a good question, and also the other part of your question that it's pretty interesting is the what happens in the in-between. And we have captured and this is a question that Tara has brought up before as well. Like we have captured the end state of these states of health that we have identified, right? That are pretty final. There's a transition there, and in some cases, when we're looking at those states of health, we're looking also at, well, is what we're finding there responsible for the problem, or is it something that showed up later as a degrader or something that is just benefiting from a dead plant, right? So that's something that we have to look more, and that was part of the idea of doing that long-term study with Josh. But again, we're picking samples at very specific times and not necessarily in a nice, long, continuous way. Brett.
I just wanted to respond to Sina's question. We don't want to be killing weeds so our lawns will grow. We should be killing our lawns and letting the weeds grow. That's the sustainable solution to that whole issue.
I was wondering why it was
just wanted to throw that out there.
I was wondering why it was taking you so long to get to that. Nick.
So it's kind of connected to Nate's question. How much do you know about the stability of the communities, or I guess the communities you study, right? So ecologically, when we think of a community, some of them are more stable than others, and when you remove a player, it's either fine or it collapses. So some of those players might be more important than others, and that might be a part of the transition. Do you know if that's the case? And the ones that you work with, obviously they're going to be
I'm just saying how much time. I'm just saying how much time we have. So for the native communities, we have found that over time they're very stable. So if we sample the same dune that is already formed at the beginning of the summer, at the end of the summer, the community changes like most communities do, but that the key players are there, right? The changes that we see are more dramatic are in the early stages of the dune formation. So when we look at a plant that is just starting to colonize and sand is starting to accumulate and that environment is changing because how deep the root is buried is changing, so all of that is going to change levels of oxygen, and that makes sense, right? So that's where we see most of the changes in the stability of the community. The part that we are a little bit more puzzled is the changes that Amber is seeing in the experiments that we have in these reconstructed communities. So this is the idea of, okay, now that we know everybody and we have genomes of everybody and we can track everybody down for a while in a setting, let's do this. We're not tracking we don't have to go to the beach and sample continuously. We can do this experiment up in the greenhouse and look at a community as it's evolving over time. And it's very dynamic. And of course, part of it is because we are putting massive loads of those microbes that it's not the way that it exists in nature. So we're just trying to see how they interact with each other in close to their original environment. But there are some negative interactions in there. There are some guys that take off as winners. There are organisms that are being, like I pointed in that slide from Amber, that we've been seeing some organisms be affected by the plant more than others in a positive way. So there is a lot of dynamics in there, but it's, again, that's a very artificial experiment. It's more to understand what kind of interactions are there and if those inocula could work as something that could be commercialized at some point. Charlie.
I'm curious of the state of the field with respect to the technology. So you think about the beach grass as microbes, members that you're identifying for use as augmenting fertilizer.
Mm-hmm.
How much do you think about identifying known microbes and using them in that capacity versus the need to identify new microbes from technology transfer perspective, right?
So the biggest challenge in there is understanding the effect on public health, right? So what do you do with these microbes, particularly for something like beach grass? If it's something that people are going to put on the beach, that you're going to use as a biofertilizer on the beach, that's something that you want to know that it's safe. I want to know that it's safe for me, my children, everybody that is going to that beach. Everybody is going to the beach. So I think that's a big hurdle, knowing that those organisms are safe. And there's ways of testing them, and there's ways of also making sure that what we're working in the lab, they are safe for everybody working with them. But it's a longer process than some organisms that are going to be used for agricultural purposes and again, those also have to pass that test. But we know a little bit more about how to make that happen quickly. And by we, I mean, not me, industry, right? So the Bayers that took over Monsanto's and AgBiome and companies like that are, getting bigger and bigger, but developing those specific solutions to crops and to other places. And that also drives the idea of trying to isolate microbes that are already there. Kind of back to Sina's point, that the idea would be to, even in this, getting this organism out of their environment, that it could be a potential pathogen, so we have to test it, but it's an organism that came from that particular environment and might make sense to bring it back if it has some benefit for that plant. But yeah, the industrial paths for making this happen are getting more impressive and more impressive. They have, we're doing very basic work in the lab compared to what they do in industry in terms of accelerating those things. But that's why something like that final slide that I was showing with the beachgrass work, that possible partnership with Join, they have approach to make that happen relatively quick, commercialize that very quick, get through all the testing that needs to be done. I am good, and my lab is good at going out there and casting a broad net and picking the things that might be interesting. They can take it to the next stage. Jess.
So, can I go? One last question? So building off of Nick's question, and then also what you mentioned, when you're inoculating these plants at the greenhouse, you're putting lots and lots of bacteria on them. So I'm curious, with your molecular work, you can see what proportion of the community, say, falls in a particular genus. Can you tell how many individuals of a particular species or strain are present? Because it's one thing to be present and you have 5 individual cells there. It's another thing to be present and you have 1,000 individual cells there. So in terms of that and how that might change through time, I'm wondering what you know and if you can even know that.
You can. So the techniques are, when you're using DNA sequencing and 6-ness DNA sequencing, they're quantitative enough to give you a sense of the proportions, relative proportions to one another. It's still based on PCR, right? So there's some bias to specific groups that are going to be more, some of them are going to be amplified more than others. But there are ways of, going back and doing qPCR, which is something that we're doing, being specific to, okay, I want to see how much that organism has grown quantitatively, and with a control, right? So we can do that, and the techniques are there. They've been there for a while. So, and the other part of it is something that, and you should have been in our environmental micro class this morning. We were discussing a paper in which, there's the other part of it, right? That one thing is that the organism is there, right? So when I was saying, asking you about how many are here, yes, that's how many sequences of those microbes are there, but it doesn't tell you anything about which ones are actively growing or actively doing something, right? So that's another thing that we can also track down, and it can be done. And that's something that Amber is doing right now with, we're doing mostly with RNA sequencing. Yeah. Yep. Good questions. Thank you all very much.